Dr. Nicholas Ray again. As we heard yesterday, he's a senior lecturer in the Institute for Environmental Sciences at the University of Geneva, and he's heavily involved in a research unit that is involved in environmental modelling and geoprocessing in order to provide data for environmental management. However, he has another string to his bow, which isn't evident in his biography, which is that your background must be in genetics and biology. So he's going to talk to us this morning from that background. So Nicholas, thank you very much.
Thanks a lot, Hilary, for this introduction, and really thanks again for the privilege of being able to talk for a second time during this wonderful week. Yesterday we looked together at the future, and we're going now to look at the past. And let me also start my talk with this nice composite view of the Earth, which this time shows you the light emitted at night by human activities. And actually the amount of light can be used in some places as a proxy for population density. Not everywhere, but in some places. But it's also, I guess, showing us how successful our species has been in colonizing this planet, in reaching the ends of the world. And of course, some of the questions that have fascinated many people is about how we managed to achieve this colonization. When did we colonize a different part of the world? How did we do that? How was the size of our past ancestors, the size of the past populations? And so we're going to look at some of these questions today. What are the type of data that can help us answer these type of questions? Well, there are several types of data. Paleoanthropological data, of course, the remains, the bones that we found of ancient hominids, ancient species, can help us understand our evolutionary links and other things about our ancestors. Archaeological data, of course, looking at the remains, the tools, the settlement of past populations, can be very informative on the timing and the type of culture and the type of technical skills that our ancestors had. Other type of data are so-called past climatic and past paleobotanical data, and paleozoological data that we find, and that will help us actually to build maps of past vegetations, helping us to understand what was the environment, and also at what time did some of these locations, some of these locations on the Earth were open for colonizations for our ancestors. So of course, with the different level of sea level in the past 1,000 years, for example, 20,000 years ago, sea level was about at 120 meters below actual level. So it really helped our ancestors to go to the Americas through the Bering Strait. We're coming back to that later. Linguistic data can also be of some help to understand the relationships between population, and there is strikingly correlations between the different type of languages and the genetic diversity in many populations. Of course, with linguistic data, we can only go back a few thousand years, 6,000 years, when we had the first writing, the first documented writing that helped us to understand the languages. And of course, more recently, I mean, in the past decade, we have genetic data that are helping us a lot to understand more about the diversity of our species, the link between the different populations in the world, and that can help us also really to live in the past and to understand what happened. So what I would like to do in this presentation is just briefly talk of humanity. And also talk a little bit about possible interbreeding between modern humans, Homo sapiens, our species, and other subspecies. And finally, I'll just have a few slides on possible models for interfacing the science in that field and the genetics account. So quickly, probably as a recap for most of you, the genetic data we have, they are composed of cells with nuclears in each of these nucleus. We have our genetic material, composed of a very long stretch of DNA molecules. I'm sorry, there's a little bit of French in here, but let's cope with that. Very long stretch of DNA that are composed of so-called nucleotide molecules, or base pairs. This is this well-known ACTG letters, four different nucleotides, put together these very long DNA stretches. The nucleus is not the only organelle in which we have DNA. We have mitochondrial DNA. Mitochondria are these small organelles that are linked to all the energy in our cells. And there are kind of stretch of circular DNAs, many copies of it in each of the mitochondria. And this mitochondrial DNA is found in much higher quantity than the DNA in the nucleus. It has been used quite a lot and still being used by geneticists to understand the diversity of species. It's also DNA that mutates more rapidly than nuclear DNA, so it's also useful for some studies. Our genome is the ensemble of our DNA material. We have 23 pairs of chromosomes, with a special pair of chromosomes, which are the sexual chromosomes, is XY for males and double X for women. Well, if I go back to this, just a small sequence of DNA here with these letters, what we call mutations are changes in these DNA sequences. There are different types of mutations. You can have a mutation that will just change one of these nucleotides. And there are mutations, for example, that are called microsatellites, where you have a small pattern of 3, 4, sometimes 5 of these letters that are repeated many dozens of times. And one mutation in this microsatellite is the removal or the addition of such a small stretch of DNA. And so there are other types of mutations, and they all have the utility in answering different type of questions. So that's really the material that population geneticists are using, so various methods to understand and to answer many questions on various species. And the DNA sequence that is coding for protein is typically called a gene. So with that in mind, just if you look at some figures, 3.2 billion nucleotides is the size of the human genome. That's quite a lot, but that it's not the size that makes us human. This fish here has a genome of 130 billion nucleotides. And this nice little flower here, I think that's the record with 150 billion nucleotide in a genome. Another important figure to keep in mind is this 1.5%. 1.5% of our genome actually code for proteins. And the rest of our genome has some functionality, and we more and more starting to understand this functionality. There are some non-coding RNA genes, regulatory sequences, and other things. But it seems that there's a lot of our genome that has no functionality. We are, as I said, diploid individuals. We have 2 copies of each of our chromosome. Some species have more. This is the so-called Viscacha rat is a tetraploid species, so 4 copies of each of these chromosomes. And strawberries, the strawberry that we eat, are octoploid, so there are 8 copies of each of the chromosomes. A lot of DNA. You know, maybe as you can do DNA cocktails with strawberry. A lot of you've done that, I guess, with your DNA yesterday as well. Just to put that in perspective. So back to our genome, 3.2 billion letters that, you know, printed in small prints that would fit all these numerous volumes here. Actually, it's lacking chromosome number 1, which would be an extra stack. And just to put that in perspective, if you would have to type 8 hours per week the DNA letters quite quickly, it would take you about 50 years to write all this. But it takes only 8 hours on average for a cell to duplicate. It's quite marvelous machinery. So what are the questions that can be addressed by genetics and genomics? Genomics is the study of when you have the full scope of the genomes and just not only a few sequences at some locations. Well, you can compare. One approach is really to use genome from different species or different subspecies, and then you can compare that and answer questions linked to the evolutionary linkages between these species, interbreeding, so whether or not there's been some mating events, hybrids between different individuals or different subspecies, selection event, whether you have been some adaptations and selection of a particular gene variant in populations. You can also compare individuals in a given species or population in a given species, and then you have a lot of statistical methodology that helps you to find out whether there has been demographic growth in the past, bottleneck, so a kind of crush of population size, population isolations, and for how long, secondary contacts, routes of migrations, level of immigrations, emigrations in populations. Population geneticists are still really improving a lot of statistical methods based on genetic data to find this type of answers in our genomes. So just a word on the theory of evolution. I know we'll have some other talks this afternoon. I guess it's important to keep that in mind. So the theory of biological evolution really focuses on the comprehension of the processes that are responsible for the past and current biological diversity. So strictly speaking, the theory of evolution is not concerned by the emergence of life. It is concerned with what happened after the emergence of life. And it's on its basic form. It's quite easy. It's really based on 2 principles. First, that there is a generation of diversity in our genomes through mutations, through combination, gene flow, etc. So there are diversity all the time. And then natural selection that operates at all time. Natural selection is simply put that genomes that will have a small advantage for survival or reproduction, will tend to be the ones that need to be passed to the next generation. And to illustrate that, with a kind of well-known graph, but really basically, if you have a population with species that with some diversity, here diversity of the height of the individuals, then well, in that very basic simple case, some of these individuals under particular environmental circumstances will have small advantages over the other individuals. And that therefore it might be the individual that will survive and that will pass on the genes to the next generation. If you continue this process for many generations, then slowly but surely the population can acquire new traits and can change the composition of the population. So that's very, of course, it's many times more complex than that. But and there are other factors for speciation and for the changing populations, epigenetic changes, symbiotic relationship, developmental genes that can have a huge effect in a small amount of time. So there are many things, but basically, just to give you a talk on natural selection. So you can often get the question, I had the question, well, especially by my Christian friends, are they really proof that the theory of evolution is true? Well, I guess that reflects a misunderstanding of what theory is in science. Of course, like all scientific theories, it's provisional, incomplete, susceptible to changes, brutal additions. And so, really in the theory of evolution, there are elements that are verified by multiple lines of evidence, very long history of life, transformation of population and species through time, unique origin, common descent, we'll come back to that. But there are also elements that are debated. The importance of natural selection against other type of generation of diversity, survival mechanism of micro-mutation, another example. And there are unknown mechanism at the moment. Again, the origin of life, that, again, strictly speaking, could be outside of this theory of evolution. Mechanism for transformation of forms, dimension of organisms, are not well understood yet. And so, I guess when you've been asked this type of question, you have to engage in a conversation to make that understand that it's not answered by yes or no to that question. It's really, I think we mentioned this image of a puzzle where we have area in the theory of evolution that works very well, we have a lot of information, the area more sketchy, but this is how the theories work. So back to human evolution. For a long time now, many scholars have tried to reconstruct the link between different species, and in the past, of course, only based on biometry, on morphological elements, on skeleton, trying to see how different species relate based on bones. And of course, recently we've been able to use genetics and genomics to do similar type of work and to try to see how different species relate. And here you have a big phylogenetic tree, so you cannot read that, but just to tell you that actually the length of these branches in this tree here relates to the amount of genetic differences between the individuals. And actually humans are right here, close to apes and others. And all the blue here are bacteria. So you can see that. And on a side note, actually, linked to my talk yesterday, you can wonder why when we talk about biodiversity loss on this planet, we never talk about bacteria loss in terms of bacterial species. There are a lot of them. So this is just, again, I don't want to spend too much time on that. Some of the hominid species that have been discovered so far, the material is very sketchy. It's very difficult to find this type of material. But we have some of them. The oldest one about 6 million years ago. Just to show you that we have here this brown color, the Australopithecus family with the famous Lucy, 3.5 million years ago. Australopithecus afarensis. And then as we move on to the close time, then we have Homo habilis, Homo erectus, Neanderthalensis that we'll talk about later, and humans. And of course, there's been attempted to try to link in evolutionary linkage these various species or subspecies. And of course, when you discover a kind of new type, it might change a bit how you sketch that type of graph. And yeah, just to say that our closest known subspecies is the Neanderthal, and we'll have a look at that later. Just quickly some word on common descent, because of course this is also a subject of debate sometimes in our church circles. Do we have evidence for common descent, for linkage between humans and other species? Well, I say we have overwhelming evidence of this. First of all, there are similarities at the level of genetic sequences and amino acid sequences between species that are closely linked. The order of genes on the chromosome is almost identical in many close species, and the order of gene apparently has no influence on how these genes are used. So this is also a good indication that they're coming from common ancestors. And also, I guess one argument when you look at our closest species, which is chimpanzee, we have 98.7 DNA similarity between our genome and the chimpanzee genome, and even 99.4% if you only look at the protein coding sequences. Well, the chimpanzee has 24 chromosomes, and we have 23 chromosomes, so they have one chromosome more. But what we discover actually is that the chromosome 2, there are two small chromosome 2 in chimpanzee, and we have only one big chromosome 2 in humans. But if you look at this chromosome more closely, so this would be the human chromosome number 2, and these are the two small 2A and 2B from the chimpanzee. Actually, what you can see, and these bands here are, if you dial the DNA certain matters, you obtain these different bands that are linked to the density of DNA along the chromosome. So the bands, as you can see, look exactly the same, but what is really striking is that you have special sequences at the end of the chromosome, those telomeres, and you can recognize that very easily. And actually here, you see in our second chromosome, very special sequences that are actually telomeres here. And what really happened is that we think there's been a fusion of these two chromosomes at one stage to give chromosome 2 in humans.
The same gene sit on the same chromosome as the chimpanzee?
Yeah, exactly.
Not just two out of them.
Sorry?
Out of the chromosome, not just number 2.
No, in that case it's just, yeah, I mean, the other chromosome mapped to our chromosome, but it's only with chromosome 2 that we have an apparent fusion of these two. So if we can sketch that, it really seems that, well, between chimpanzee and humans, we have a common ancestor that is think to have lived maybe 6 million years ago. And then an evolution branches one up to the chimpanzee and other branches to humans. But at one stage, during this evolutionary history, then we have this fusion of these two chromosomes. That's the story that you can really tell from that. I guess there's a really strong argument to say that, well, if God would have created kind of de novo humans with absolutely no linkage with other species, why would he have put that kind of very proof of fusion right here that is not functional in our chromosome 2? There are other, of course, evidences, and there are thousands of them. Certain type of viruses can insert themselves into our genomes. And actually, we found more than 100,000 retrovirus sequences in our genomes. That's quite a lot. 5 to 8% of our genome, of your genome, is made of virus sequences. Quite amazing. But what is amazing is that, of course, these viral sequences are found in the exact same place in genome of many related species. That's really an indication that this insertion has taken place way back in the past, before some kind of splitting of species, and that we found these evidences right now in our genome. And the word of Denis Alexander is not with us, I guess. We are really living museums of our evolutionary past. There are thousands really of other examples, pseudogenes, jumping genes that we found at the exact same place in different species, and I guess, proof of our common descent. Now, just let's jump to geographic and temporal origin of humanity. There's been alternative models that have been discussed, and are still discussed. The main one is the so-called out of Africa model, where at one stage Homo sapiens evolved from Homo erectus in Africa, and then get out of Africa and colonize Asia, Europe, Australasia, and then really the source of the expansion is sub-Saharan Africa. That's the typical simple way of putting the out of Africa model. And the multi-regional model has been discussed a lot in the end of the 20th century. It's now not really followed, but this model postulated that actually modern humans evolved simultaneously in Africa, in Asia, in Australasia, in Europe. So from different Homo erectus populations, but for a much longer time period, for millions of years. And due to, and they had to invoke a lot of migrations between these different areas in order to keep a kind of global diversity. And then these different populations of Homo erectus would independently evolve to modernity in these different areas. And that was based mainly on morphological features of the different skeletons that were found in Asia and other places. Now the models now that receive more ground is the so-called assimilation model, where we keep the South Africa model, but in which you have to take into consideration interbreeding, so assimilations, mating events between modern humans and other type of humans. What are the arguments in favor of this unique origin, of this unique geographic spread and recent spread of humans? Well, the global population, human population, has a relatively low diversity, genetic diversity, which is indications of shallow past in terms of evolutionary history. The highest diversity is observed in sub-Saharan Africa. Again, that's an argument to say that currently living sub-Saharan African populations are the descendant of the most ancestral populations of our species. And the genetic diversity decreases as you move away from Africa. And you can really do a correlation between the distance from, let's say, Eastern Africa and the genetic diversity of full population in the world. And you have a correlation of about, if I remember, 0.9. So it's really very high. So this decline from Africa is really a sign of this reduction of diversity as you go away. It's a really well-known process in genetics. That's an indication that there have been this expansion from Africa. This is a graph that shows the putative routes of migrations of humans from Africa and the kind of rough age of these different events. So modern humans emerged in Africa between It's really difficult. You cannot actually say when that happened, because speciation is really a process. It's the population that evolved. It's not the individuals. It's very important to consider. And so it really seems from the fossil records that there have been quite long transitions between very archaic modern humans to late modern humans. So we cannot say exactly where that happened, 150, 2,100 years ago, emergence of modern humans. Then 50,000 years ago, the dramatic events of the out of Africa. It's very well known in the archaeological records. Two possible routes, still disputed. Then colonization of the rest of the world. It's even possible that we have found remains at 70,000 years ago in Australia. And that would mean that actually there potentially be an earlier out of Africa with very quick migrations along the coastlines to Australia. And they've made the trip, and even with the very low sea level, there were still, I think, a couple of, or 300 kilometers of sea to reach Australia. And our ancestor made that trip several times. You know, there was no sails at that stage, so they made it, so that was quite amazing. And then colonization of Europe, actually, latest data is now around 45,000 years ago. And the latest major event is colonization of the Americas through the Bering Strait, about around 15,000 years ago, again, when the Bering Strait was emerged, 1,000 kilometers wide, a lot of animals on it, so we think they probably followed some herds of animals and found themselves in a new continent. What is really interesting is that recently we have indirect biological evidence of human migration, looking at other organisms that were tightly linked to humans. That's the case of this bacteria, Helicobacter pylori, that is famously known to be linked to stomach ulcers. And scientists have been sampling this bacteria in all population of the world, sequencing the genome. And actually what they found is that the diversity of this population, the structure of the diversity of this population closely follow the structure of the genetic diversity of humans. So, and they've been evolving also, of course, independently, the genome evolved independently of the genome of humans, but they have been evolving also with the timing, a kind of split that our past humans have also encountered. So it's good, yeah, it's another type of evidences and way of understanding how the past populations have split and evolved in the past. And if some people have done the same work with human lives, there's been less evidence here, but it seems that so far, it's been published in 2013, the genetic diversity of these beasts here also closely related, also resemble the kind of genetic diversity structure that we have in humans. So another indications and alternative line of evidences of how the population colonized different parts of the world. Let's have a look now at this interbreeding with other subspecies. Neanderthal, this is a reconstruction from National Geographic. You can notice the red hairs here, and it's because when they sequenced the genome, they found specifically the gene that is responsible for red hairs in the genome of Neanderthal. So it doesn't mean that all Neanderthals have red hair, but at least that this particular trait apparently existed already. This is the distribution, the different sites, and probably they are missing a few now, different sites in Europe where we found remains of Neanderthals. And that's the putative distribution of Neanderthals 50,000 years ago, so just prior to the entrance of modern humans in Europe. So modern humans arrived in Europe about 45,000 years ago, and that's from some very recent paper published last year that have really looked at all the body of evidences from archaeology and genetics, and especially on looking at the debated timing at which Neanderthal disappeared from Europe. It's now apparently clear that they disappeared about 40,000 years ago. So there has been 5,000 years in which we cohabited with Neanderthals in Europe. This is just some graphics showing you the difference in morphology between Neanderthals and humans, reconstruction of faces. So probably if Neanderthal now would have modern clothes in the street or on a rugby field, you wouldn't recognize it. So, of course, the major question between Neanderthals and modern humans is whether or not there's interbreeding. If so, what is the proportion of our genome that is coming from Neanderthals? And were there any advantage or disadvantage of interbreeding for our species? So that's the type of question that we can now answer, and that there are now several studies looking at that. In 2006 was the first breakthrough where a team, international team, managed to sequence some of the genome of the Neanderthals. They started with mitochondrial DNA and then they went on with nuclear DNA. It's extremely, actually, or it was at that time, extremely difficult to sequence all the DNA, so ancient DNA. Many problems, and one of which, of course, is contaminations. Because we have all DNA on our fingers. If I touch anything, there's DNA on other things. And so archaeologists that were manipulating the bones are really like sponges. The DNA of the archaeologists will just be in the bone. And so you're pretty sure that if you sequence anything, you are sequencing the DNA of the archaeologist. So it's extremely difficult, and they have to go through very difficult things to avoid contaminations, and statistical methods were developed to kind of also correct for contamination and many other things. 2010, the first full draft of the Neanderthal genome, and that, of course, they compared to our genome, and what they found. Well, they found that what they found when they looked at several genomes, several individuals, Neanderthal individuals in Europe. But the best explanation of what they found is that actually outside Africa there is a 1 to 4% contribution of Neanderthals to the modern genome. That really means that on average, people outside Africa, and in Africa it's not the case, outside Africa, have 1 to 3% of their genome that is much more likely to be linked to a Neanderthal ancestor. So we are, so that was the finding at that time. That was not the case in African population. And so really what they found out of this is that this interbreeding, this mating event, of course, occurred when the modern humans out of Africa expanded into the territory of Neanderthals. That's why we could not find this contribution in contemporary African population. And there have been many more studies since 2010. The field is advancing very, very fast, and that have confirmed that. Actually, I found this paper just before coming here, published in 22nd of June. And the same team, so Sven Paper from Germany, usually the leading scientists in that field, what they managed to do is to sequence a node modern individual dated between 37 and 40,000 years ago in Romania. And they found out that actually in his genome, 6 to 9% of his genome was derived from Neanderthals. And they computed that actually, and there were very large chromosomal segments from Neanderthals. And they concluded that this would be possible only if this individual had Neanderthal ancestors, I think 6 to 8 generations in the past. recode. And so we have more and more of this material, and it's quite amazing. Again, we can really count on many new discoveries in the future. Yeah, so 4th to 6th generation back. Was there the effect of interbreeding on our fitness? There are also interesting other studies published recently in 2014 that compare genomes between Neanderthals and modern humans. And these teams converge in their finding, and they found out that actually, apparently some European Asians gained genes from Neanderthal that was involved in skin and hairs, and they postulated that maybe it helped our ancestors to adapt more rapidly to colder climates in Europe and Asia. Thicker skin, more hair, fewer pores. But they also found out that there were also some other genes that make us more susceptible to, for example, type 2 diabetes, Crohn's disease, and others. So I think we're just at the beginning of understanding of the potential, advantage or disadvantage of interbreeding our fitness. And I guess that's also food for thought for theologians. This type of findings. Another immensely interesting discovery was the Denisovans, that we found remains of these unknown humans in a cave in Siberia in 2010. A tooth and a phalange were found, dated back at 40,000 years ago, extracted the DNA, sequenced the DNA. What they found is that it was neither a modern human, neither a Neanderthal. They call them Denisovans. And what's striking, what they found is actually in all the populations, current populations in Melanesia and Papua New Guinea and northern Australia, Aboriginal populations, well, there were 4 to 6% contribution in these populations of this Denisovan. And so, yeah, it really seems like that's another type of subspecies, that there's an interbreeding event. And again, the conclusion was, the model so far is that as modern humans were colonizing Eastern Asia and then Melanesia, they encountered these other populations that we don't know exactly what it was, probably what's ranging somewhere here, and then interbreeding events. And as the modern humans continued the colonization, they kind of took over, spread these genes that they took from this Denisovan, and it's now visible in contemporary populations. So the global picture so far, and probably also we are missing some very recent event, is that modern humans went out of Africa, encountered Neanderthals with several interbreeding events, and probably there's more than 2. Then went on to Asia, encountered also another type of populations, Denisovans, and continued to wait. And there are now speculation about a mixture with archaic other type of humans in Africa, and it's very likely that in the coming years we'll find other events out as well. So we think that it's quite sketchy. It's not as simple as we saw. It's the model that would summarize all this is more like really an intertwined bunch of events rather than simple out of African model. So we've seen this, well, just to go to the final stage of my presentation, we have on one hand data, scientific theories that are based on observations, have been shaped through time. We have theological truths that are within the scripture, and some of them you can consider as eternal, non-refutable. Of course, as was well presented this morning, we have many interpretations of scripture that can also change through time. But I think it's perfectly fine to try to, for yourself, to have a model that tries to integrate all this into something coherent, that describe how scripture and science cohabit. And what I would like to do right here, and I guess that's maybe helpful for our discussions and maybe to try to position yourself if you're not done so, is to present six models on the origin that were portrayed in the book of Gerald Rowe in 2012, called Mapping the Origin, the basics model of the beginning of everything. And I think that maybe quite useful if we just go quickly through it. And after that, I'll present a special model that actually was put forward by Dennis Alexander that I particularly like, and I'll tell you why. So the first, so Rowe is really trying to span the variety of models that typical evangelical Christian, I would say, would have, starting from well, and more than evangelical Christian, of course, in that case, naturalistic evolution, so the general idea is materialistic evolutions with only natural processes. So of course on Genesis it's an ancient myth, there is no God, no supernatural, no purpose, no intervention, no problem of course with common descent, with an old universe, and that would probably be the position of atheist or agnostic. Then, so number two, non-teleological evolution. You know, in that model, conditions necessary for life are established at creations. So Genesis would be an ancient myth, but God exists. There's a creator, but there is no purpose, no intervention, and again, no problem with common descent and no universe. So again, that could be for different types of monotheism, theism, and others. And then there's the so-called planned evolution. Again, kind of theistic evolutions, where you have a kind of perfect creation that naturally fulfills the purposes. There is no concordism. So concordism is we try to seek the harmony between the geological record and the days of Genesis. We talked about that. Adam and Eve are individuals. So historical Adam and Eve, creator. There is a purpose with that model, of course, but there is no specific intervention by God. And again, it's okay with common descent into the whole universe. And the fourth model would be the directed evolution, where actually the changes in the universe and the evolutionary process would be directed over time by the creator. No concordism, Adam and Eve.
Concordism, what does it mean?
Concordism is when you try to see the parallels between the geological record and the days of Genesis. So I guess we talked this morning that some people will try to see the first day, that's what happened. The second day, that's what happened, and try to fit that to what we know about evolution. Again, in that model, it's okay with common descent, all universe, and that's a monotheist approach. Now, the all Earth creation model or perspective is really trying to do concordism, so really trying to match what the Genesis account says and what the evolutionary process is, but with somehow some extended days, so to allow for all universe. So the creator, of course, purpose, interventions of God in that model, and yeah, that's a position that is held by some people, and of course the intelligent design somehow is part of that. And then finally, at the end of the spectrum, we have the young Earth creationist, recent creation, 6,000 to 10,000 years ago. Each guy is created on its own. So it's really taken a literal view of the days in Genesis. Creator, purpose, intervention, creation de novo of each species. So no common descent. It's a recent creation. So just to give you an overview, and that might be useful, but what I would like to do in my final two slides is really to put forward the model that Dennis Alexander in his book Creation of Evolution, Do We Have to Choose? And I would really recommend that book if you're interested in that subject. It spans more than human evolution, but it's really up to date. The latest edition is 2014. And Dennis is presenting, I he had 5 different models that somehow overlap a bit with what Rao is presenting here. But he has a particular model he calls Homo dividus model that I particularly like, and I'm particularly happy to consider when I personally try to consider what the science is telling us and what the scripture is telling us. In this model, so the content of this model is a proto-historic model. It makes reference to events that took place at particular time and locations. Events localized in the culture and geography described in Genesis. What about the first spiritual humans? Well, in that model, God at one stage choose a couple or a group of Neolithic farmers. So the Neolithic is the period where the agriculture started, so 10,000 years ago, 10 and 8,000 years ago. So God chose a group or a couple of people from these Neolithic groups in the Middle East and reveals himself, reveals to them in a particular manner. And that's really the start of the very special relationship that humans have with God. So there is the consciousness of God in the core that started at this moment, while, of course, all the humans were living everywhere on the planet. The Americas, the Australia, not Antarctica, but on the other continents. So that's where the start of the authentic spiritual life starts. Of course, in that type of model, you have to invoke a kind of federal chief of humanity that is in Adam, or in this initial group of people. And of course that had implications for the rest of your model. Argument for that model and implications, well, for the cultural context of Genesis resemble that of the Neolithic farmers, agriculture, metals, etc. It's really in agreement with the existence of the humanity that has lived at that time. The text suggests that other humans were present, and we discussed that in Lance this morning, so yeah, the biblical genealogy, I don't want to do a lot about that, but we agree with that time frame. In that model, the flood, again, is probably a historical event, but yet a very a local event. And there's absolutely no trace of a global flood, not in the genetic record, not in the archaeological record. And finally, well, in this model, the fall is the disobedience of Adam and Eve leading to this spiritual death. But again, the difficulty in that model is you have to somehow cope with this federal chiefness of humanity that is in Adam, because his fall must have somehow imply the rest of humanity, but how exactly that, the process of that. So that's probably the one of the theological difficulty for that type of model. There is a kind of natural paternity and not only by descent. But again, theologians have to jump on that and discuss many issues. So I guess I'll finish with that model and I'll thank you for your attention.
Thank you very much, Nicholas. Plenty to think of there. Actually, I will just tell you the story of someone I met earlier this year who is himself a geneticist and paid to have his own DNA sequenced just out of curiosity because he's a scientist and wanted to know. He actually found out, slightly disturbingly, that he had a higher than average genetic tendency towards Alzheimer's, which wasn't something perhaps he wanted to know. But he also found that he had a higher than average proportion of Neanderthal DNA. And if I say he had teenage children, you can imagine the kind of fallout from that in the family discourse, which was quite amusing. But anyway, let's take some questions on that. Yes, Mike. It should be on.
Could you comment on the non-functional? So you said
said, for instance, that I think it was 98.5% of the DNA doesn't code for genes. Is that correct? Something like that? Doesn't code for proteins.
Yeah, it's 1.5% that
And I can remember teaching about this 20 years ago. We used the term junk DNA to something.
Yeah.
Do you remember that? And you also said that the DNA has no function. Would you like to comment on that? Because I would question the word no function.
Yeah, you're right. I'm not really a specialist of that particular part of the genome. You're right in saying that junk DNA nowadays wouldn't be used anymore because we've learned so much about the DNA structures outside the genes, and we found out more and more that indeed some of it has functions on many things. And so, but there are still yet, and again, I cannot tell you the percentage outside genes that is considered so far as non-functional, or we haven't found out yet. But certainly, I would say that, that there is structures that might be completely non-functional. And I will add that this also should be seen as protection, because if you would have only a much smaller genome that would be only functional, you have much higher risk of having mutations that would fall into important genes and that would have detrimental consequences. And so I guess that's somehow a gift that we have so much of the DNA that is non-functional. And actually, it's a gift for population geneticists because we're using, of course, the mutation in outside the genes to tell things about the relation between populations, which is harder to do when you look at mutations in genes because they could be subject to selections, and there are many issues there.
That's the fact now.
That's the function that helps scientists work things out.
Sorry?
That's the function that helps scientists work things out. You just,
Oh, yeah.
So,
I'm a little curious about what the implications of our current migratory patterns are for future research. All of that is based on populations that stay in a pretty small area for a pretty long time. And that's not the case anymore, obviously. Are we getting more diverse or less diverse because of the greater intermingling across space? Is there anything you can comment on that?
You mean the intermingling of human populations nowadays?
Mm-hmm.
Yeah, that's a good question. I have a slide. I'll just show you something. Because that's an interesting point. Yeah, this nice map shows the contemporary connections between populations. So you have all the flights, you have the roads, you have the trains, you have the boats. Really show that there's a lot of movement in contemporary populations. And actually that has been a problem for population geneticists because of course when they are sampling population in Australia, in North, South America, in order to trace back what the evolutionary history, they want to make sure that they have people that are not coming from Europe or that have grandparents that come from Europe, because of course you're looking at the genetic diversity of Europeans rather than local populations. So that's why you always have to look at, if you can, kind of very or hunter-gatherers population that you, or Amerindian population that you think are descendants of the ancestral population in the continent. But nowadays it's almost impossible to find population that, especially in South America, that do not have a portion of the genome that comes from Europeans. And so that has led to the development of very advanced statistical methods to take into account this, and you can actually now have this method that allow you to look only at the genetic diversity outside the European parts of the genome. But I think your questions was related to whether, you know, how our genetic diversity is changing right now. Well, I guess I would say that, well, there's a lot of changes, but it is really recent changes, and that doesn't change really the kind of overall human diversity. You can still, you know, take many people and really looking at their diversity tells exactly where they come from the earth, because we have this very essential diversity that you know, keeps with us and will keep with us for a long time.
Okay, just in front of you, Colin, there.
Yeah, I like, as you do, Dennis's model and this idea of God revealing himself to a special group of people in the Fertile Crescent, while at the same time there were other humans around in the rest of the world. I think there's a bit of a danger, though, in assuming that those other people in the rest of the world had absolutely no relationship with God at the same time. And I think we see a sort of extrapolation of that in modern times, because I heard particularly one sermon preached in a church, I won't say where, when the preacher got up and proclaimed that anybody who had not had the gospel of Christ preached to them by a missionary or someone was therefore damned to hell. And so many were going there, clicking his fingers every minute around the world, because they were the unreached. And I don't think it's our place as humans to say whom God has reached or not reached, or whom God has, or who has reached God or not reached God. So I'm wondering if, and I haven't read Dennis's model in detail, but I'm wondering whether there is any discussion or any idea that while this special revelation was being made and everything that flows from that, there are not in other parts of the world some kind of relationship between Homo sapiens and God.
I mean, that's a big
Otherwise, it's very exclusive, isn't it?
Yeah, you're right. Yeah.
I have an idea that may be a question that's addressed this afternoon. I'm not sure, but it may be. But actually, I believe also there is paleoarchaeological evidence for certainly things like ritual and burial rites and things like that, which are happening in other parts of the world, which suggests some capacity of humans to at least envision another being, some sense of religious awareness. So I think it's an interesting question, isn't it, Nicholas? Yeah. Just Adele.
Yes. Sigmund Freud said that God was a genetic idea. Do you have any thoughts on that?
Yes. God is a genetic idea?
Yeah.
What does that mean?
Well, probably, well, it could be related to, you know, some of the discourse that says that there is an evolutionary advantage in believing in God, or a type of God, and that, of course, that it evolved as any other trait that evolved, because it helped population to, you know, to survive better than if we didn't have that. You know, of course, that's not I believe, but I know that some evolutionary biologists are taking that route and are trying to you know, find some proof of that. But I couldn't comment more than that.
Thank you.
In the middle.
Yeah, I've got a basic
Can
question.
you hold it right
Sorry.
up close?
Yeah. A basic question really that I guess admitting I'm confused about the definition of species now. I think it's a true fact that I as a Homo sapien can only breed with other Homo sapiens. Now, is that something which is unique to in human history? And is it unique? I mean, are there other primate species today which can breed in the same way we could breed with Neanderthal?
Well, you're right. The definition of species has been quite evolving, and it's this, of course, the sort of strict sense of species, which is, you cannot interbreed. Now it's being a bit reviewed because we know so much more about, hybrids and how things can happen, and it's rather continuous of, possibility of hybridization than rather strict barriers. But yeah, you're right also in saying that if our ancestors have interbred with Neanderthal, but strictly speaking, Neanderthals were not another species. And so of course nowadays, well, apparently yes, we cannot interbreed with other species, but yes, I guess in our past, with apparently those so many different subspecies that we have, probably it would have been a continuous process of for some times. Maybe some of the population, the Neanderthal population, would not have been able to interbreed because of biological barriers. Some other would. Probably we were in a continuous of possibilities. And now, as for the today apes population, from what I know, in the higher apes there is no interbreeding possible, so there are different species. But yeah, it's the question of species and interbreeding is many types of animals. We see things now that are more of a continuous type of things.
Okay. Thanks.
Kamal?
Thank you, sir, for your marvelous presentation. I have a simple query which is related to this slide also. My query is human migration. There was another slide you have shown, human migration. Could please open. Yes. This slide you have shown that mostly African people are going outside. Migration is very frequent, and here one thing that is United Kingdom has not been no arrow there. I think recent news published there. The most still the mostly tourist destination is UK, but it is quite totally ignored. I don't know what is really. No, people usually come to UK and some of them are residing and making their home. So I need some clarification. Thank you so much.
This is really a basic sketch of in the past, long time ago, on English people, but you should also really be cautious actually when you see this type of arrows, because they definitely do not reflect reality or kind of, that doesn't mean there have been 3 events or that they follow that route. We really cannot trace back the routes that they have followed, because we have some data, we can have some idea about the timing, but often we have quite high uncertainty. We can make assumptions about the routes and the motifs, but really it's just you have to be really cautious when you see that type of thing. The only thing is that I was involved in the study that showed that there has been 3 major migrations to the Americas. The first one being responsible for all the kind of Amerindian genetic diversity. The second one apparently related to some Canadian populations, and the third one the northern parts. But apart from that, we have to be very cautious. It's very simple.
Okay, we've just got time for one more question. Okay, I'll take George. Sorry, Nathan. George. Yeah.
Sorry, just approaching this from a physicist's point of view, what sort of error bars are there on some of the dating? And also, going back to the speciation point, I know from a geological point of view, a lot of the species stuff is done on morphology, which now is looking to be a little bit suspect, a little bit dodgy perhaps, in drawing those barriers and grouping the species and things like that. And have, from a genetics point of view, have, in retrospect, these various discoveries then being genetically profiled to try and work out if they are actually separate species or just odd morphologies or and the like.
Yeah, thanks for the question. The error bars on the archaeological material is I would say quite low, probably in that range of value with sometimes a few thousand years. So to be quite sure, because the error bars on the timing that is using genetic data, genetic reconstruction, is much higher. And this is just because of the way the phylogenetic reconstruction works. It's, and those error bars are could be quite high. And we have been involved in studies where, we have a kind of posterior distribution of the timing. So you have the best estimate maybe at 70,000, but the error bars goes from 120 to 30. Of course, with the new, this is also a field that advances very quickly, and there are really each year new statistical methods using Bayesian approaches and all that can really trying to track that, but we cannot do that beyond a certain point because it's just the nature of how these genes are transmitted from one generation to the other. And I would say again about this speciation time, it's we don't know. It seems to be a continuous process. In a morphological record, it is a continuous process. And genetic data here at the moment cannot help in pointing, a certain time for the speciation. Certainly, again, speciation is a long process for
Great. Well, I'm sorry we've got no more time for questions, but let's thank Nicholas again.